Hello and welcome dear students. Today we are going to discuss the topic post-translational modifications. The main objectives of this module are to know about the process of translation, to discuss the process of post-translational modifications, to understand various types of post-translational modifications, to know the location of post-translational modification in the cell, to understand the importance and application of post-translational modifications, and finally, to understand the role of post-translational modifications in regulation of gene expression. Dear students, let's start with the process of translation. The DNA of the chromosomes located within the nucleus contains the entire store of genetic information. Selected sites on the DNA are transcribed into pre-mRNAs, which are processed into messenger RNA. The messenger RNA formed is transcription carries genetic information encoded as a ribonucleotide sequence from the chromosomes to the ribosomes. The ribonucleotides are read by translational machinery in a sequence of nucleotide triplets called codons. Each of those triplet codes for a specific amino acid. The ribosome molecule translates this code to a specific sequence of amino acids. In translation, mRNA is decoded in a ribosome outside the nucleus to produce a specific amino acid chain or polypeptide. The polypeptide later folds into an active protein and performs its functions in the cell. This process of gene expression is used by all known organisms and can be regulated and modulated at several levels like transcription initiation, splicing, alternate splicing, mRNA stability, post-transcriptional regulation, and eventually translational and post-translational regulation mechanisms. Dear students, let's now understand the post-translational modifications. The function of the protein depends on its 3D conformation. The 3D conformation of the protein in turn depends on the interactions between amino acids in the polypeptide chain. Following translation, translocation or insertion into endoplasmic reticular membrane, proteins are modified to assume their final structure and therefore function. Post-translational modifications change the chemical nature of the polypeptide chain through alterations to amino acid residues. Proteins are modular in nature and their interactions with other molecules in the cell rely on the presence of specific functional domains. The precise shape of the domain resulting from the presence of non-covalent bonds between residues in a polypeptide chain decides about the function. Translation in biology is a process in which a molecule of messenger RNA is sequenced to amino acid during protein synthesis. Post-translational modifications refer to the changes occurring to the protein with the help of enzyme called protein synthesis. Post-translational modifications involve modifications of the amino acid chain, terminal amino or carboxyl group by enzymes. Due to these modifications, the structure, stability or activity of the proteins get affected. The processes which cause modifications are phosphorylation, methylation, glycosylation, nitrosylation, ubiquitination, acetylation, lipidation and proteolysis, to name a few. Such modifications happen as crucial molecular regulatory mechanisms to regulate diverse cellular processes. These processes have a significant impact on the structure and function of proteins. Disruption in post-translational modifications can lead to the dysfunction of vital biological processes and hence to various diseases. Dear students, let's try to understand where do the post-translational modifications occur in the cell. Post-translational modifications take place in the endoplasmic reticulum and include folding, glycosylation, multimelic protein assembly, and proteolytic cleavage, leading to protein maturation and activation that take place as soon as the growing peptide emerges in the endoplasmic reticulum and is exposed to modifying enzymes. Only properly folded, properly modified proteins are transferred to Golgi and secreted through the process of exocytosis out of the cell for secretory proteins or merging with the plasma membrane for membrane proteins. Misfolded or unfolded proteins accumulate in the endoplasmic reticulum and cause upregulation of molecular chaperones through the endoplasmic reticulum overload response. If chaperones cannot fix the problem, defective protein would be transported back to cytosol, ubiquitinated, and destroyed in proteasomes. Post-translational modifications take place also in cytosol. Some of them, for example, phosphorylation can happen repeatedly throughout the life of the protein and are the frequent way of regulating protein activity inside the cell. In doing so, each protein becomes prepared for its specific and unique function in the cell, taking into the account local environment conditions at the final destination place. Dear students, let's now discuss some key points of post-translational modification. Post-translational modification play a key role in numerous biological processes by significantly affecting the structure and dynamics of proteins. Generally, a post-translational modification can be reversible or irreversible. The reversible reactions contain covalent modifications and the irreversible ones which proceeds in one direction include proteolytic modifications. Post-translational modifications occur in a single type of amino acid or multiple amino acids and lead to changes in the chemical properties of modified sites. Post-translational modifications usually are seen in the proteins with important structures or functions such as secretory protein, membrane proteins, and histones. Post-translational modifications affect a wide range of protein behaviors and characteristics including enzyme function and assembly, protein lifespan, protein-protein interactions, cell-cell and cell matrix interactions, molecular trafficking, receptor activation, protein solubility, protein folding, and protein localization. Post-translational modifications are also involved in various biological processes such as signal transduction, gene expression regulation, gene activation, DNA repair, and cell cycle control. Post-translational modifications occur in various cellular organelles including the nucleus, cytoplasm, endoplasmic reticulum, and Golgi apparatus. Dear students, let's now discuss about the various types of post-translational modifications. There are more than 400 different types of post-translational modifications affecting many aspects of protein functions. But according to the type of modification, these post-translational modifications can be categorized into three main groups. First and second groups are those post-translational modifications that include the addition of chemical and complex groups to the target residue respectively. The first group and the second group include glycosylation, prenylation, myristoylation, and palmitoylation. Those post-translational modifications that contain addition of polypeptides to the target residue comprise the last group, and these post-translational modifications are ubiquitination and sumoylation. Dear students, let's now discuss some of the important and major post-translational modifications in detail. First, the glycosylation. Protein glycosylation is one of the complex forms of post-translational modifications and is formed by the addition of carbohydrate molecules to an amino acid to form a glycoprotein. Carbohydrate chains are added to proteins in a reaction called glycosylation, which occurs in the ER or Golgi. Glycosylation is further divided into two types. That's N-linked glycosylation, where a sugar molecule is attached to the amide nitrogen of asparagine, and O-linked glycosylation, where sugar molecules are attached to the oxygen atom of the serine or threonine. Glycosylation is a characteristic modification for secreted and extracellular proteins and consists of the addition of one or more carbohydrate side chains that change the chemical properties of an amino acid, thereby creating space within or surrounding the molecule. Glycosylated proteins are also important and clinically relevant opportunistic receptors for viruses and toxins to enter the cell through receptor-mediated endocytosis. They are also common cell surface antigens, of which ABO blood types are probably the most known example. A unique glycosylation attachment of mannose-6-phosphate has a double duty for lysosomal proteins. It works as targeting signal for lysosomal enzymes and then protects them from being digested. An N-linked glycosylated protein acquires its carbohydrate chain as a preformed oligosaccharide in the endoplasmic reticulum, and an O-linked glycosylated carbohydrate chain is added in the Golgi complex. One sugar at a time. N-linked glycosylation starts as soon as glycosylated residues, always asparagine or arginine, makes it to the lumen of the endoplasmic reticulum as a nascent peptide, which is still unfolded. A preformed oligosaccharide is then transferred from an ER membrane-bound lipid carrier dolicol to the polypeptide chain. O-linked glycosylation that occurs in Golgi during later stages of protein maturation is catalyzed by sugar-specific glycosyl transferase enzymes. Sugars are transferred from the nucleotide precursors. This process is important in the formation of extracellular matrix proteins. Dear students, let's now discuss the second post-translational modification, phosphorylation. Protein phosphorylation was first reported in 1906 by Phoebus Levine with the discovery of phosphate in the protein vitellin. However, it took another 20 years before Eugene Kennedy described the first enzymatic phosphorylation of proteins. Phosphorylation is the reversible attachment of the phosphate group to a specific residue of the target protein. Phosphorylation is catalyzed by protein kinase and phosphatases. Phosphorylation is important for cell growth, cell cycle, and signal transduction pathways. Protein phosphorylation is the most studied post-translational modification and can be used in disease processes such as cancer. Phosphorylation plays a key role in the activities of many enzymes, membrane channels, and many other proteins in prokaryotic and eukaryotic organisms. Phosphorylation target sites are serine, threonine, tyrosine, histidine, proline, arginine, asparagine, and cysteine residues. But this modification mainly happens on serine, threonine, tyrosine, and histidine residues. It includes transferring a phosphate group from adenosine triphosphate to the receptor residue by kinase enzymes. Conversely, dephosphorylating or removal of a phosphate group is an enzymatic reaction catalyzed by different phosphatases. Phosphorylation is the most studied and one of the essential types of post-translational modifications, which often happens in cytosol or nucleus on the target proteins. This modification can change the function of proteins in a short time via one of the two principal ways, by allostery or by binding to interaction domains. Phosphorylation has a vital role in significant cellular processes such as replication, transcription, environmental stress response, cell movement, cell metabolism, apoptosis, and immunological responsiveness. It has been shown that disruption in the pathway of phosphorylation can lead to various diseases such as cancer, Alzheimer's disease, Parkinson's disease, and heart disease. Dear students, let's now discuss the third type of post-translational modification. It is the ubiquitination. Ubiquitination is one of the most important reversible post-translational modifications. This modification was first studied in 1975 by Gideon Goldstein. The modification is a versatile post-translational modification and can occur on all 20 amino acids. However, it occurs on lysine more frequently. This post-translational modification has a major role in the degradation of intracellular proteins via the ubiquitin proteasome pathways in all tissues. In ubiquitination, a covalent bond befalls between the C terminal of an active ubiquitin protein, a polypeptide of 76 amino acids, and N terminal of lysine residue of the protein. Ubiquitin can occur on mono or polyubiquitination forms on substrate proteins through specific isopeptide bonds by receptors containing ubiquitin binding domains. Ubiquitination is catalyzed by an enzyme complex that contains ubiquitin activating, that's E1, ubiquitin conjugating, that's E2, and ubiquitin ligase, that's E3 enzymes. Unubiquitinated proteins may be acetylated on lysine or phosphorylated on serine, threonine, or tyrosine amino acid residues and lead to dramatically altering the signaling outcome. Ubiquitination modification in substrate proteins can be removed by several specialized families of proteases called deubiquitinases. Ubiquitination plays important roles in stem cell preservation and differentiation by regulation of the pluripotency. Ubiquitination regulates many processes such as cell cycle, cell growth, functions of the immune system, membrane exchange, DNA restoration, transcription. Ubiquitination has also played a vital role in many various cell activities such as proliferation, regulation of transcription, DNA repair, replication, intracellular trafficking, and virus budding, the control of signal transduction, degradation of the protein, innate immune signaling, autophagy, and apoptosis. Dysfunction in the ubiquitin pathway can lead to diverse diseases such as different cancers, metabolic syndromes, inflammatory disorders, type 2 diabetes, and neurodegenerative diseases. Dear students, let's now talk about the fourth post-translational modification, that is the acetylation. The first acetylation modification in proteins was discovered by V.G. Alfray in 1964 in isolated calf thymus nuclei in vitro. Acetylation is catalyzed via lysine acetyltransferase, that's CAT, and histone acetyltransferase, that is HAT enzymes. Acetyltransferases use acetyl-CoA as a cofactor for adding an acetyl group to the alpha amino group of lysine side chains, whereas deacetylases or HDACs remove an acetyl group on lysine side chains. There are three forms of acetylation: N-alpha acetylation, N-epsilon acetylation, and O-acetylation. N-alpha acetylation is an irreversible modification, and the other two types of acetylation are reversible. These three forms of acetylation occur on lysine, alanine, arginine, asparagine, cysteine, methionine, proline, serine, threonine, and valine residues with different frequencies, although the acetylation is more reported on lysine residue. And alpha acetylation is more biologically significant compared to other types of acetylation. Acetylation has an essential role in biological processes such as chromatin stability, protein-protein interaction, cell cycle control, cell metabolism, nuclear transport, and actin nucleation. According to the available evidence, acetylated lysine is vital for cell development, and its dysregulation would lead to serious diseases such as cancer, aging, immune disorders, neurological diseases like Huntington's disease and Parkinson's disease, and cardiovascular diseases. Dear students, let's now discuss about another post-translational modification, that's methylation, in detail. Research on methylation dates back to 1939, but with the identification of new methyl transferases such as protein arginine methyltransferases or PRMTs and histone lysine methyltransferases, that is HKMTs, has attracted more and more attention. In protein methylation, the methyltransferases enzyme adds a methyl group to the specific amino acid on a protein molecule like lysine and arginine residues. Protein methylation is effective for protein stability, subcellular localization, binding affinity, and protein-protein interactions. Methylation is a reversible post-translational modification which often occurs in the cell nucleus and on the nuclear proteins such as histone proteins. Methylation occurs on the lysine, arginine, alanine, asparagine, cysteine, glycine, glutamine, histidine, methionine, phenylalanine, and proline residues in target proteins. However, lysine and arginine are the two main target residues in methylation, at least in eukaryotic cells. One of the most biologically important roles of methylation is in histone modification. Histone proteins, after synthesis of their polypeptide chain, are methylated at lysine, arginine, histidine, alanine, and asparagine residues. An epsilon lysine methylation is one of the most abundant histone modifications in eukaryotic chromatin, which includes transferring the methyl groups from S-adenosyl methionine to histone proteins via methyltransferase enzyme. In eukaryotes, methylated arginine has been observed in histone and non-histone proteins. Recent studies have shown that methylation is associated with fine-tuning of various biological processes ranging from transcriptional regulation to epigenetic silencing via heterochromatin assembly. Defects in this modification can lead to various diseases such as cancer, mental retardation, Angelman syndrome, diabetes mellitus, lipofuscinosis, and occlusive disease. Dear students, let's now discuss about the another post-translational modification, SUMOylation, that is small ubiquitin related modifier, that is SUMO protein, was primarily discovered in 1996 by Rohit Mahajan in the RAN GTPase activating protein, that is RAN-GAP. SUMOylation takes place via SUMO that has a three-dimensional structure similar to ubiquitin protein and has been discovered in a wide range of eukaryotic organisms. SUMOylation can occur in both cytoplasm and nucleus on lysine residues. SUMO family has 3 isoforms in mammals, 4 isoforms in humans, 2 isoforms in yeasts, and 8 isoforms in plants. SUMOylation occurs as a modifier in alpha amino group of lysine residues in target protein through a multi-enzymatic cascade. In this reaction, SUMO is connected to a lysine residue in substrate protein by covalent linkage via 3 enzymes, namely activating, that is E1, conjugating E2, and ligase, that's E3 enzyme. Also, it is separated from the target protein by a specific enzyme, protease SUMO. SUMOylation plays a major role in many basic cellular processes like transcriptional control, chromatin organization, accumulation of macromolecules in cells, regulation of gene expression, and signal transduction. It's also necessary for the conservation of genome integrity. Also, there are many reports on major role of SUMOylation in development of a variety of human diseases including cancer, Alzheimer's disease, Parkinson's disease, viral infections, heart diseases, and diabetes. Dear students, let's now discuss another post-translational modification in detail, that is lipidation. An important class of post-translational modifications called lipidation includes covalent attachment of lipids to proteins. The first report of the covalent modification of proteins with lipids dates back to 1951. By lipidation, proteins are targeted to membranes in organelles such as Golgi apparatus, mitochondria, vesicles, and plasma membrane. These post-translational modifications are taken place via a great variety of lipids like octanoic acid, myristic acid, palmitic acid, palmitoleic acid, stearic acid, cholesterol, etc. Myristoylation, palmitoylation, and penylation can be considered as the three main types of these lipid modifications. There are four types of lipidation: C-terminal glycophosphatidylinositol, anchor, N-terminal myristoylation, S-myristoylation, S-penylation. GPI anchors, which are made in the endoplasmic reticulum, are added to the new protein block. GPI anchored proteins are localized to cholesterol and sphingolipid rich lipid rafts that act as signaling platforms on the plasma membrane. N-myristoylation gives proteins a hydrophobic handle for membrane localization and is catalyzed by N-myristoyl transferase and uses myristoyl coenzyme A as a substrate to attach the myristoyl group to N-terminal glycine. Proteins that undergo this post-translational modification play critical roles in regulating the cellular structure and many biological processes such as stabilizing the protein structure, maturation, signaling, extracellular communication, metabolism, and regulation of the catalytic activity of the enzymes. The role of myristoylation has been proved in the development and progression of various diseases such as cancer, epilepsy, Alzheimer's disease, Noonan-like syndrome, and viral and bacterial infections. Dear students, let's now discuss another post-translational modification, that is proteolysis. This process includes limited and highly hydrolyzed peptide and isopeptide bonds, protease, ubiquitinaceous and thermodynamically favorable and irreversible modification. Proteolysis is a regulator mechanism and its activity can be controlled by proteolytic induction. Proteolysis is a thermodynamically favorable and irreversible reaction. Proteases are broadly divided into four categories: serine proteases, cysteine proteases, aspartic acid proteases, zinc metalloproteases. Dear students, let's now talk about the role of post-translational modifications in diseases. Post-translational modifications have a vital role in almost all biological processes and fine-tune numerous molecular functions. Therefore, the footprints of disruption in post-translational modifications can be seen in many diseases. Neurodegenerative diseases is the major group of diseases which is affected by the disruption in the post-translational modification, like Alzheimer's disease, Parkinson's disease, and Huntington's disease. Besides, one can see that cancer is also one of the most affected disease. Consistently with this observation, the biological process related to cancer are among the high degree nodes, that is signaling, DNA repair, control of replication, and apoptosis. Processes related to apoptosis, protein-protein interaction, signaling, cell cycle control, chromatin assembly, organization and stability, DNA repair, protein degradation, protein trafficking and targeting, regulation of gene expression, and transcriptional control are the other high degree biological processes. Moreover, we can say that ubiquitination, prenylation, glycosylation, S-palmitoylation, and sumylation have the most involvement in diseases. On the other hand, the post-translational modifications with the highest number of interactions with biological processes are phosphorylation, ubiquitination, methylation, acetylation, and sumylation. Putting all together, we can conclude that the disruption in the pathways of these 5 post-translational modifications has a great impact on the normal functioning of the cell and as a result on the organism. Dear students, let's now conclude the lecture. Post-translational modification of proteins being one of the later stages in protein biosynthesis refers to the reversible or irreversible chemical change proteins may undergo after translation. In other words, post-translational modifications are chemical modifications of a polypeptide chain that occur after DNA has been transcribed into RNA and translated into protein. These chemical alterations range from the enzymatic cleavage of peptide bonds to the covalent additions of particular chemical groups, lipids, carbohydrates, or even entire proteins to amino acids side chains. These chemical modifications of a polypeptide chain after its biosynthesis extends the range of amino acid structures and properties and consequently diversifies structures and functions of proteins. Post-translational modification can occur at any stage of the protein life. Some proteins are modified shortly after their translation is completed and prior to the final steps of their folding. These early post-translational modifications might affect the protein folding efficiency and protein conformational stability and even determine the fate of the nascent protein via directing it to distinct cellular compartments. Other proteins are modified after their folding and localization are completed. Here, post-translational modifications can activate or inactivate catalytic functions or otherwise influence the biological activity of the protein. So, post-translational modification of proteins plays some significant functions in cell growth, cell process, etc. The post-translational modification of proteins are important for controlling the stability of the protein, localization, and conformation. By multiple post-translational modifications, genetic information imbibed in DNA is transcribed, translated, and increases its complexity. Post-translational modifications help in the mediation of stress perception, protein homeostasis, energy shift control, and defense by the immune system. Dear students, it was all about today's lecture on post-translational modifications. Hope you have understood it well. See you with a new topic next time. Take care. Goodbye.